Melanoma Matters is an educational resource. The content represents our opinions and does not constitute medical advice. If you're a patient with questions specific to your situation, please consult your medical practitioner. Hi everybody, welcome to Melanoma Matters. I'm James Larkin.
And I'm Sapna Patel.
And we're honoured today to have with us Jedd Wolchok. So, Jedd, I don't think you really need any introduction, but nevertheless, it'd be great if you could say something about your background, where you were, maybe why you got into oncology in the first place before we sort of start talking a bit about CheckMate 67.
Sure. So first of all, James and Sapna, thank you very much for the introduction to be part of this wonderful broadcast that you have. And I think it's a really great opportunity to share our knowledge with colleagues and the wider community as well. Like many people, I entered the field of oncology because of family members who were affected by cancer, and sort of recognized the importance of developing new therapies. And just by happenstance, I was actually introduced as a university student to a team of physician scientists who were investigating melanoma. That would be Alan Houghton and Lloyd Old at Memorial Sloan Kettering in the mid-1980s. And they were investigating antibody-based therapies. In that era, state of the art was a fully mouse antibody to a ganglioside, GD3, on melanoma cells. And I had the real privilege of being the entire PK department for a phase 1 clinical trial. So that just tells you a little bit about how the world has changed, that a 19-year-old college student would be the entire PK division for a phase 1 clinical trial, where now there are probably teams of investigators who are actually paid for their work, clinical companies. But nonetheless, it was a phenomenal introduction to truly translational research, how things move from the laboratory to the clinic and back again. And it really was one of those moments that affected really the rest of my professional life, because I saw how science could intersect with medicine in a very direct way. And it inspired in me the desire to really do both of those things when I grew up, which I'm not sure that I ever have, but actually, I've embraced this border between science and medicine, and bringing things from the laboratory to the clinic and back again.
Jedd, you were 19 when you met these profound individuals that sort of changed your trajectory. Can you tell us where you did your education and training, maybe for the audience, who Lloyd Old ended up being to you, and then this trajectory to being Cancer Center director?
Yeah, so at the time I was an undergraduate student studying molecular biology at Princeton, and Lloyd Old was the vice president of research at the Sloan Kettering Institute. Alan Houghton was a young physician scientist in Lloyd's group, then breaking out on his own as an independent laboratory investigator, but also the head of the melanoma service at Sloan Kettering. I went on to do my medical training at New York University, and then my internship at Bellevue Hospital, and then went to MSK, Memorial Sloan Kettering, as a medical oncology trainee, stayed there for a short 25 years after that, and then 2 years ago moved to Weill Cornell to become the director of the Meyer Cancer Center. For those listeners who are not in New York, that geographic move was not very difficult. I'm fully about 15 feet away from where I was before. So really not a difficult geographic move.
Oh, that's great.
Yeah, and so if I remember correctly, Sapna, that Jeff Weber spent some time at Bellevue as well.
At Bellevue. as well. He worked in the emergency room there. He was not sure yet of his course, and so he took some time, he said, in the emergency room and said he had amassed many a good story in that
I think
few
we
emergencies.
all have amassed great stories, taking care of people from every walk of life, from every corner of the planet that,
Yeah,
fortunately trusted us at early points in our career with their care.
yeah, that's
Yeah,
great.
so maybe before we talk about Checkmate 67, I mean, you were involved with IPI really from the earliest days. And this is a sort of personal recollection of mine. I mentioned this to Sapna the other day that I think when you presented the phase one IPI-NEVO, it was ASCO, I don't know, maybe 2013, something like that.
Your memory is very accurate, James.
Well, what I was going to say though is that at the time, you know how you have those evening meetings sponsored by industry, and you'll go to one, and there'll be some food and stuff like that, and then you show the data and talk about it. Well, I remember extremely clearly. I was at one of those meetings, ASCO 2013 in the evening, when you came along and you presented the phase one data, and incredibly striking. I think that's what I took away, and I thought, wow, this is something that we need to be doing.
Yeah, it was a very special moment when we realized that even though both of the different approaches to T cell, modulation, whether it be CTLA-4, PD-1, although they converged on the T cell, they got there by very different ways, and so there was value in trying to bring them together. And it was a really fantastic opportunity to work closely with some really great colleagues on that phase one trial. As you might recall, it was a 2-institution trial between Yale and MSK. And actually, we had the great fortune, I would say, of having Jeff Weber and Steve Hody as the data safety monitoring committee.
Mhm.
So we had real partners in forging a path ahead for what some people might have considered to be a fairly outlandish, idea of bringing together two potentially toxic medicines in a meaningful way. And the team, at least the team from BMS, as well as Mario and Harriet from Yale and myself and colleagues from MSK, we would talk every week about every single patient. And we really were, it was an interesting time because we really had to present this data at the same time that the monotherapy activity for Nivo and Pembro in melanoma was emerging. And so there was, a lot of necessity of demonstrating that there was something different going on with the combination. And so as you know from Checkmate 67, we looked at the data, like upside down and sideways, different PD-L1 expression cutoffs, different, different mutational backgrounds, BRAF mutated versus wild type, to try to understand a little bit about, where the sweet spot was for the development of the combination. But you're right, James. It was 2013 when I presented the phase one data. Was actually shortly thereafter that I was invited to a dinner, which I didn't really know the magnitude of, down in Princeton, New Jersey, where the headquarters of Bristol Myers Squibb is, and to meet with their senior executives. And really, the purpose of that was to sell the idea of of Checkmate 67 to higher management at the organization, because what we had actually done, myself and 3 trainees that I had the real honor of mentoring, Mike Postow, Maggie Callahan, who were both at MSK at the time, and then Arvin Yang, who had finished his training and went to BMS. They were 3 of my, most wonderful, colleagues. And they, along with myself and actually, I believe Omid Hamid was also there. We were standing in the lobby of one of the ASCO hotels, and we were saying, well, what's the next step, for the phase one? What do we need to accomplish? And so we thought about a phase 3 trial that was essentially a contribution of components, 3-arm trial, and we sketched out CheckMate 67 on the back of a cocktail napkin. And I wish I had saved it, to be honest with you. Now, the big difference was that we wanted a crossover design to the combination for either of the monotherapy groups where there was progression. Sure. That was unfortunately not taken forward because of concerns around contamination of the overall survival endpoint.
Right.
But when we finished dinner that night, one of the executives at Bristol Myers, at the time Michael Giordano, pulled me aside and said, "You got your phase 1 trial. You got your phase 3 trial. You got the big trial."
Amazing.
So, and then, identifying James as my partner in this adventure, we really moved forward very quickly, and the trial accrued rapidly. And kudos really to the teams all over the world that accrued this trial in a very rapid fashion.
Yes, Sapna, before we go on, I was going to say something before we go on to you.
Of course.
Those were lovely comments about your trainees, and I have to say, I'm embarrassed to say I didn't know Arvin Yang had trained with you, because I only knew him when he was at BMS.
Yeah.
And maybe not for now, but we worked really closely together actually with the regulatory submission in Europe, because the EMA was not as straightforward as the FDA had been, and Arvin was just fantastic in making that happen. And I still have some contact with Michael Giordano today, actually. He's moved on. I don't know if that's a whole different story. Anyway, Sapna, back to you.
Well, that was actually wonderful context. That was exactly what we were hoping to hear, which is just like the kind of, how did this come about? Was this really, and it sounds like it was really experienced investigators. And then, you had to keep it small at first, but then you said, I think this is a real synergy with these two compounds, and we have to think big about it. And so that has led.
There was so much learning. Sorry to interrupt you, but one of the things that came out of it, which actually Mike Postow, took in a very important direction, was the dose escalation from the phase 1 trial was actually supposed to, go quite a bit higher. And we explored 3 milligrams per kilogram of both drugs. And the only thing that stopped that was a preponderance of grade 4 asymptomatic lipase elevations, which turned out to be completely biochemical in nature with no accompanying symptoms of pancreatitis. And Mike really took that on as an example of, the tail wagging the dog, that we need to incorporate the symptomatic meaning, the clinical relevance of laboratory values that we may encounter, especially when they are influencing the way that we conduct clinical research. Now, I'm not saying that IPI3 Nivo3 is any better. We'll never know that, right? But we do know actually that IPI10 is better than IPI3, right? And so, at least for the ipilimumab component, there might have been an opportunity to go higher, but we had to respect the rules of the road.
Yeah. Well, I said this on an earlier podcast. And by the way, we did discuss your work with dose escalation in some earlier episodes, because we come to that often as melanoma oncologists. We think about dose responsiveness of certain drugs, and then other drugs where, a single dose can sit on a receptor and it's not as dose responsive. And we're sort of arbitrarily often maybe capping drugs, or maybe not exploring the full range of their potential. But we think of your dose escalation when it was Metarex, and certainly then when it turned into ipilimumab with BMS. One of the comments I made was that Patrick Hu had told us, as you mentioned, you guys were in very close contact, you and the Yale investigators, as you guys were running the phase 1. And of course, that the word of that trickled out. And so Patrick came and said, look, these agents are really active. We know that from the other trials, but in combination, they're extremely active. Mario is telling us if we use this combination, we have to buy every patient a new toilet. And that is such I mean, obviously he was saying it tongue in cheek, but he said, look, we really have to be careful. And what happened was at the time, MD Anderson was in more of a mindset of we don't do big phase 3 trials. We do the grassroots investigator initiated. We participate in phase 1s or we help do phase 2. So we don't need to do phase 3s. But he made that comment around August, and we had passed on CheckMate 067 because we felt like we didn't need to be site 200 on that. And then I brought it up. I was very junior at the time, and I said, I recognize the academic reason why we said no to it, but isn't there a real world component here, which is we all believe this is going to be a real regimen that we will use. And is the first time that we use it, do we want it to be after it's approved? Because when we study things on a clinical trial, there's often really nice oversight, guardrails for toxicity. You can learn because your monitor and your colleagues and everybody is helping you. Once it's approved, you have a prescribing information document to guide you. You don't have any necessarily experience. Based on that, we changed our mind and we said, let's go all in with 067. Let's do it. Let's participate. I think Patrick and Wenjen said, Sapna, then you take it on. So we did, and we opened it in late November or middle November. We were open for 4 days, put 4 patients on, and then it closed globally. So, I mean, this really speaks to how quickly you guys did accrue this study, how popular it was. And then also it was just a reminder to us that it's important to participate in these things. Just if you see the new wave is coming, certainly we wanted the experience. But maybe that's all talking about where we are kind of then, and we should get to where we are now. Jedd, we would love your take on the data, maybe the high points as you see it. We heard James give us 4 minutes and 59 seconds of his take on it. So we know his high take on it. And then I did ask him at one point that I'll ask you, have you ever seen a New England publication with punctuation in it? This says final, comma, 10-year outcomes. I'm not sure I've ever seen a comma or a question mark in a New England title before.
So for the last point, I was sort of struck by the definitiveness of it. But maybe it was just sort of like, the New England Journal, we were very fortunate. They published this clinical trial 3 separate times.
Yeah.
With different points obviously being made. But I think it really was a testament to, what can we learn by following the same group of people for over a 10-year period of time who, especially when they started this treatment, when 2 of the 3 groups were experimental, right? Nivolumab had not been approved. Certainly the combination had not been approved. Ipilimumab had been approved for a fairly short time. So there was a lot that we had to learn. I can't add too much to James's very eloquent presentation of the primary data, but at a high level, I think what it told us were a few messages. First of all, the way that the curves flatten out, for lack of a more scientific term, after 3 years, gives us a lot of confidence in the way that we interact with patients and their families after that point. That we really, and this was hinted at in the article, right? Can we start to use the word cure? And some of the post-publication, social media, et cetera, said, if this doesn't represent cure, then really what does? So I think that 10 years of follow-up show us that, yes, there are a few people in whom, the melanoma will subvert the immune system and come back, but it's really very few, especially in the people who show a greater than 50% reduction in tumor burden early on. So those were a couple of the points really mashed into one observation that, first of all, we talk about complete and partial responses. Those are not definitive categories. There are plenty of people in this particular trial, at least, who had residual abnormalities on their imaging. Those in whom half of the abnormality was still present at the end. that still have not shown, death from melanoma. And so it makes us wonder how much of what we're looking at on this imaging is viable tumor versus not. And thankfully now in 2024, we have other means of assessing outcome in addition to, of course, the gold standard of cross-sectional imaging. We can think about things like cell-free DNA as the lower limit of detection becomes better defined as supporting some of these conclusions. I think all of us in the melanoma field have had patients who've asked, well, can we take out whatever is residual? And at least in my own experience, when we've done that exercise, it's been debris that's been identified, and it's not been viable tumor that we found. So I think that's one observation, that not every residual area of abnormality is cancer. The second is that we really can, I think, back off after 3 years in terms of repetitive imaging. And I think that's important for patients in the quality of life, area, because they can start to really get back to a normal list of concerns and not like the every 3-month scanxiety, which is a real thing. And in addition, to a point that was made earlier, I think Sapna, you made this, is when do you stop treatment? Well, the truth is when you're in a clinical trial and there are rules that dictate when you have to stop treatment based upon toxicity, what we see again here in this trial is that stopping treatment early, and that's an artificial, definition of early because it's made up, for the purposes of a trial, how long someone gets treated for. If the immune system is activated to the point of creating toxicity, then, job may be done. You might not need to give treatment. And that has profound ramifications at a societal level, right? Patient doesn't have to come in for infusions anymore, expose themselves to additional toxicities, or, to stress the healthcare system in terms of the cost of medicines when they might not be necessary.
I mean, you bring up points there that this type of data and long-term robust follow-up tells us, first of all, we need the marker for when the immune system is activated against cancer and has actually generated the memory cells, because we have all had patients who get an effector T cell response, and certainly if you stop for toxicity or other reasons might actually have progression because we didn't give enough to get memory T cells. So what is our tool, our test, our marker to tell us memory T cells have developed? What's our radiographic tool to tell us that this is metabolically inactive lesion that the radiologist is having to call a lesion, but it's not actually, active anymore? And what is our perhaps circulating marker to say we no longer are shedding anything? These are all now tools that as we have effective therapy, we can start matching our diagnostic tools to help us try to determine some of this. So
a really, really important point. And I think that so many of these things, especially the biomarkers for what's the relevant, immune cell subset that, we should make our decisions based upon, these are still open questions.
Mm-hmm.
And I actually think, Sapna, not to ingratiate myself to you, but I think that SWOG 1801, gives us a lot of opportunity to address some of these really important questions, as will Nadina, and other neoadjuvant studies, because the tumor, resected relatively early on after initiating therapy, is going to, I think, reveal a lot of the biomarker information that we're hoping for.
Well, the love fest goes back to you and James then, because I think 067 is potentially sitting on thousands of bloods from these patients, pre-treatment, on treatment, and maybe even during survivorship from the responders, non-responders, et cetera, that also probably are a very handy tool here if we can figure out how to interrogate it. Thank you for that.
Oh yeah, if we can, reduce this to something that is blood-based, that would truly be ideal. I mean, when people sometimes ask me, what do I think the next 5 or 10 years of oncology is going to show us. What among other things like cell therapy for solid tumors, what I'm hoping is that we develop the ability to make precision guided treatment decisions, in the same way that we now can do that for signaling inhibitors. If we can do that for immunotherapies based upon ideally a blood-based tool, but that's again, fantasy at the moment.
Yeah.
So maybe I could just ask something about the sort of the longer term aspects of this. We've got 10-year data now, and I'm not going to ask you if you went back to clinic on Monday morning and did everything very differently, but maybe, the people that you are thinking about stopping imaging and maybe not seeing in clinic anymore, how do you actually follow them up? Do they get discharged? Because, you guys work in a different system from me. Do they go back to their family doctor, general practitioner? Or might you still have a phone call once a year, something like that? How does that actually work in practice?
It's a great point, James. And I think it's very patient dependent. There are some people who, they want to shed the role of being cancer patient as quickly as possible. And, if my own bias is that certainly after 5 years, and maybe for those people who have these profound responses after 3, if they have a good GP and importantly, a good dermatologist who's going to examine not only their skin, but their draining nodes, I'm very comfortable to remain, in the back pocket, so to speak. But there are some people who don't want to lose contact, and I respect that. Some institutions have developed survivorship clinics within their own oncology practice where, people will be seen by an advanced practice provider who has knowledge of the disease. Because the truth is some GPs are appropriately terrified of a patient who had a diagnosis of an otherwise nearly uniformly lethal cancer, went through an experimental therapy 10 years ago and are now deposited back in their office for primary care. So I think we approach this in a very heterogeneous manner and adapt to what the patients want. Most people, I stop the imaging certainly after 5 years, and I will see them once a year by that point. And especially if they're being seen by a dermatologist in our organization, I feel like I've got good contact with them.
Yeah, I mean, Sapna and I have talked a little bit about survivorship before, actually, in a different episode. And I think all of us around the world are struggling a little bit with this. Sometimes there's reimbursement considerations and all sorts of other things. But it sort of feels to me, not just in melanoma, but in other tumor types as well, we've got this population of patients who probably, let's say, operationally cured, and probably many of them are actually cured as well. Should they be going to one clinic? Are there some common issues that we need to think about as a group of oncologists? I don't know the answer to any of these things myself. Sapna, back to you. Anything we haven't asked that we should have done?
No, I mean, I think this is all so great. I think it goes without saying there were no late toxicities at this 10-year mark, of course. From the other prior reports, you can see that even, late toxicities, while we know they can occur. And that gets to this point about where these patients should be followed. At what point can you say we are likely clear of a late emergence of toxicity, therefore it really is fair to deposit you back in the primary office, or to have some partnership? Certainly the survivorship model is one that is spreading across cancer centers here so that they can remain at least with close touch or near touch to the oncology team. But Jedd,
agreed.
Unless there's anything else you think we missed as a high point here, I'll give you final word on CheckMate 067.
Well, I mean, I don't think we've missed anything, but I just to your last point, which is of course very important, is that we didn't see any signal of some other common problem emerging. With 10 years of follow-up, we certainly did see other cancers. We saw heart disease, we saw cardiovascular and cerebrovascular events. But, I think that the opportunity to address this melanoma-specific survival concept was another, real opportunity that I'm glad that we were able to take advantage of. I first of all thank you for being such wonderful colleagues, for inviting me to participate in this. This CheckMate 67 adventure was really a highlight of my own career, to really interact with so many wonderful colleagues from all over the world, from industry, from academia, from private practice, to all in the name, right, of bringing better care to patients with a disease that, as recently as 2010, we didn't have good answers for.
Yeah. No, that's right. Maybe I'll just make two final points, because one is this MSS, melanoma specific survival. It's pretty clear when you look at this 10-year data. I mean, there's nearly a 10% difference in overall survival and melanoma specific survival. These patients are dying of other things. I mean, this is incredible. We've had this conversation in SWOG before and offline, why the FDA would probably not look at that as a regulatory benchmark. But at some point, when we are changing the natural trajectory of the disease, can we impact their cardiovascular and their other causes of death? Perhaps not. That's a lot to put on the oncologist's office for an OS endpoint. But when you have therapy that is now showing they are not dying of their melanoma, it's something else. 70+% of these patients can be alive at 10 years. That really has to catch everybody. When we were telling patients before, 6 to 12 months was your median survival, and that's in our lifetime. It almost feels unbelievable to say it. The other thing I'll say is, by virtue of this trial that both you and James have done, and then, the hundreds of investigators around the world, obviously that is hundreds of thousands of melanoma patients' lives. But the other thing, Jedd, you've been really incredible with this work very, very early on, is you mentored people to touch it and take it and figure out their own way to use it, with Mike, with Maggie. And, that is invaluable, because then they go on to mentor people. And so the ripple effect just continues. And so, I mean, really kudos to you. James is an excellent example of somebody who gives, pays it forward in that very same way. So I think that's just amazing. You call this a high point of your career, but I think in reality, the impact you've had is so, it's like a galaxy of impact that you've made in our
Well,
field.
thank you. I mean, I think that the real thrill, right, is stepping out of the way, right,
Yeah.
and letting other people, have the opportunity to do even greater things.
Yeah.
And Mike, started to do that, right, with the adapted approach,
Yeah.
saying like, well, now we have something that can work. Do we have to be giving all of this treatment? So that's a great example. I'll also say that one of the really fortunate downstream effects of CheckMate 67 was how it influenced care of patients with earlier stage melanoma. So the combination being used in trials like Nadina, and then other cancers, right?
Yeah.
So seeing this combination move forward in renal cell carcinoma, and in hepatocellular carcinoma with similar medicines, right? And, we are thankfully at the point where treatments are not necessarily tied to the histologic diagnosis alone. That there is a true mechanistic lens through which drug development is now being viewed.
Yeah. Super.
Yeah, there were several beautiful quotes there, Jedd. Several. The real thrill is stepping out of the way. I love that.
Yeah.
I wish I could get, because I treat kidney cancer as well. I wish I could encourage publication of disease-specific survival in that data set, but that's a whole separate question, still up in the air. Anyway, Sapna, do you want to wrap up for us?
Sure. Well, thanks, Jedd, so much for your time. I think this is such a great conversation about 067, how far we've come. We are so grateful for your time today. So thank you so much.
Thank you again.
All right, I'll hit stop here.